Should be okay. Links, video, YouTube page. Yeah. Thank you. People are still coming in.
Yeah.
Where's? Do you want me to tell me when to go?
Maybe it's still not on here anymore.
Oh, really? What's your dad's name again?
Larry.
Oh, actually, do you want to log in?
No. You do? No.
Do you want me? I can't message him because I'm not friends with him. Okay. Message. Okay. It should work. Yes. Cool. Okay. Sure. I'm gonna leave it here.
Everything working?
Everything's good now. I'm gonna leave this up here.
Great, thank you all for coming. I'm going to keep the introduction really short because Lilian has lots of slides to share with us. So usually I like to give the audience, and thank you all for coming, by the way. She has lots of family and friends, an idea of the student's personality. And in the past, I've picked out superheroes, but Lilian picked this one out herself, I believe. The lab had a happy hour, and the flyer, it had a character for each lab member, and Lilian picked Princess Ada from the movie It's a Bug's Life, which I think I've seen once. And I think that this matches Lilian's personality really well. Feisty, stubborn, caring. I added extremely hardworking. She also got a master's in medicine along with her PhD. Very intelligent, responsible, perfectionist, and you're going to see that today. There's a little worry wart in Lilian too. We were going through practice talk last night. Appearance, you can, when she stands up, you'll see, very similar to Princess Ada. And I think she tucked her wings in her dress today. So she's princess of the colony and the queen's oldest daughter. And she has, in the past 6 years, made her home my lab, Monarch Lab. I feel extremely fortunate. I guess her famous quote for the movie is, "You don't think I have offended the warriors, do you?" And this reminds me of Lilian. When she first came, she would speak, but she would always add an inflection at the end, as if she was asking me instead of telling me. And now she tells me. So today, you're going to see Lilian is the queen.
Thank you so much for that, Denise. She's always been so supportive, even until the final moments leading up to today. So I'm so grateful, and I'm also so grateful for all of you coming here. And I'm really excited to share with you what I've been working on for my thesis, and this has been focused on the transmission of salmonella. And in particular, I've focused on how this is influenced by our gut microbes, so the intestinal microbiota that lives within inside of us. And so salmonella is an important human pathogen. It's a gram-negative intestinal bacteria, and it's a major cause of human disease throughout the world. So over 100 million cases worldwide each year are caused by salmonella, and this results in over 200,000 deaths per year. It's a single species, but it can actually be distinguished by different characteristics. So it's comprised of over 2,500 different serovars, and it causes a wide range of disease ranging from a self-limiting gastroenteritis, so food poisoning that we get here in the US and other parts of the world that are more developed. And this can vary where you are. So in parts of the world that have unsafe drinking water, typhoid fever is endemic here. And this is a systemic invasive disease. And this is a picture of survivors of the earthquakes that impacted Nepal earlier this year. And typhoid is now becoming an epidemic. And it's extremely worrisome because these strains are actually multi-drug resistant to antibiotics. So they're difficult to treat. So Salmonella infects a broad range of hosts, and this varies depending on the serovar of Salmonella. So some serovars are host restricted, and this would be Salmonella typhi, which is the causative agent of typhoid fever. And so this affects just humans, and this is this systemic invasive disease. So the infection begins in your intestinal tract and then spreads to other host tissues, so it goes systemic. And there are a subset of these infected patients that become supershedder carriers. So they're asymptomatic, but they continue to spread Salmonella in their feces, and this is what is transmitted to new hosts, and they're a major reservoir of the disease. And then in contrast, we have serovars such as Salmonella typhimurium, and this is a generalist serovar, so it infects a broad range of hosts. And it's zoonotic, so it can spread between humans and animals. And in humans, it causes food poisoning. So this is a common cause of food poisoning that we have here. And this is, again, this self-limited infection where the infection affects your intestinal tract, and after some diarrhea, you eventually clear it and you're okay. However, this is not the case if you're immunocompromised. So if you are also infected with HIV or another pathogen, it could spread systemic as well. And Salmonella typhimurium infects this broad range of hosts, including some that we are in contact with. So recent outbreaks of Salmonella typhimurium have been in this African dwarf frog, these tortoises and hedgehogs. These are exotic pets that we have. They also affect livestock as well as poultry, and this is why they're a common cause of food poisoning. And they also are actually host adapted to mice. So they cause a typhoid-like disease in mice, which allows us to study this in the lab. So how do we study transmission today? So we look at the number of cases. And so we can tell this is data from the outbreak that's going on in Africa of multiple drug-resistant Salmonella. You can see, different strains that are affecting patients over time. And you can see that there is an increase in some strains, such as Salmonella typhi. And you can also look at the antibiotic susceptibility of these strains, and you can tell that there's an increase in the percentage of these that are multiple drug resistant. We can also get more detailed information about this by DNA analysis. And so with advances in sequencing, we're able to define down to the single nucleotide changes in the pathogens that are causing the disease. And this allows us to determine strains that are causing an outbreak. So putting these sorts of data together, you can retrace the origins of an outbreak. So they know this multiple drug resistant isolate of typhi came from Africa and has spread from there. And sometimes you can also trace the source of an infection. So with the Salmonella food poisoning outbreak that we're experiencing now, Salmonella sushi, we're able to trace that back to a plant in Malaysia. What's harder, though, is identifying the carriers that are responsible for this disease. And we know some of the large-scale environmental risk factors that are important for Salmonella transmission. So this includes climate and natural disasters, such as the earthquake. Major risk factors also include unsafe drinking water and consumption of raw food. And then a major risk factor is contact with a carrier. These individuals are really hard to define. And we also know that not all individuals have the same infective capabilities. So some individuals might spread disease to just one or two other hosts. But however, we've learned through studies in epidemiology that the major reservoirs of disease are a minority of infected hosts that are responsible for 80% of the infection. And so these are these supershedder carriers that can be asymptomatic but shed large amounts of Salmonella. And so they're really important for the spread of the disease. And Salmonella is spread by the fecal-oral route, and infection begins when you ingest contaminated food or water. And so the infection begins in the small intestine, and Salmonella arrives here after you've consumed it. And it can then move distally to the large intestine of the cecum and colon. However, to spread systemic, which is what we see in typhoid fever, these persistent infections, Salmonella invades Peyer's patches, which are these specialized immune centers in the intestinal epithelium. And after invading or being taken up by these cells, Salmonella then interacts with a variety of different immune cells, and from there drains through the lymphatics to the mesenteric lymph nodes and spreads to the systemic sites, such as the spleen, the bone marrow, liver, and gallbladder. And then it's thought that Salmonella in the gallbladder actually recedes the small intestine through secretions in the bile. And from there, Salmonella can then pass through the large intestine to be transmitted further. And we can study this process in mice. So we actually have 20 to 30% of our mice that become supershedders, and we use a 129 background of mice. And what this means to be a supershedder is that this mouse will shed high levels of Salmonella, over 100 million per gram of feces. And these mice are also tolerant to this high pathogen burden. And what I mean by tolerant is that they're asymptomatic, yet they're carrying this high burden of Salmonella. And so this is the characteristic of a supershedder that we're interested in. However, Salmonella isn't alone in the intestinal tract. We know that it's actually in the presence of over 100 trillion members of our intestinal microbial community. And this is a high resolution image of that microbiota community in our intestines. And it's been pseudo-colored so you can appreciate how diverse this population is. They're all different shapes and sizes. They form close associations with their epithelial cells and with their diet. And we know they're important for human health and disease. In fact, we can think of them as our inner ecosystem. The idea is that sometimes we might be more microbial than human. This is just on the basis of there's 10 times more bacterial cells than human cells in our body. There's 100 times more bacterial genes and 1,000 times more species. So this is an incredibly diverse environment. And it's possible that it might play a role in Salmonella infection. And we know that the microbiota plays an important role in controlling susceptibility to infections. And this is something that's called colonization resistance. And so this is a process by which members of the microbiota take up space. It forms this dense layer in our intestinal tracts and on top of host mucus that makes it difficult for pathogens to invade and gain a foothold to establish infection. We also know that the microbiota is utilizing nutrients that the pathogen now no longer has access to. However, we know that this can be disturbed with the use of antibiotics. And so in this case, we drastically deplete the members of our own commensal community, and this gives Salmonella room and the nutrients to bloom. And this is true for Salmonella, and this is true for Clostridium difficile, another important antibiotic-associated pathogen. So we know that the microbiota plays a crucial role in controlling pathogen infection. But we also know that Salmonella has its own tricks to emerge in the intestinal tract, even if antibiotics aren't used. And so the first step of this is that Salmonella uses hydrogen metabolites from the microbiota. It does this using an enzyme called hydrogenase, and it can then use these hydrogen metabolites as an energy source and then replicate. But in order to be transmitted, Salmonella needs to replicate to higher levels. And to do this, it has these specialized virulence factors called the Salmonella pathogenicity islands, 5, 1, and 2. And these effectors induce inflammation in the intestinal tract and depletes the microbiota. And another trick is with this altered composition of the microbiota, new nutrients are produced that Salmonella is able to use to its advantage and replicate. Now, we know that this is what's happening in the intestinal tract, but its lifestyle in systemic tissues, which is what we see in typhoid fever, is different. So in systemic tissues, such as the liver and the gallbladder, Salmonella is isolated. It's not interacting with other members of the microbial community in these sites. Furthermore, Salmonella is usually residing within these immune cells called macrophages. And so this is a very different environment, and you can think of it as two different habitats. So Salmonella in systemic sites is an isolating habitat, kind of like the Antarctic, perhaps. And then Salmonella in the intestinal tract is kind of like a watering hole where it's interacting with a variety of different organisms. However, within a single host, these habitats are interconnected. Furthermore, when we think about infection of new hosts, you can think of it as invasion of a new ecosystem. And so for the first part of my thesis, I was interested in applying ecological principles to see if I can predict transmission dynamics. So given that salmonella infection begins in the intestinal tract and then spreads systemic to these sites here shown in gray, I wondered if different subpopulations of salmonella form. And by subpopulation, I mean a local group of organisms within a habitat. So this would be a subpopulation of salmonella in the spleen versus a subpopulation of salmonella in the cecum. And if these different subpopulations do form, what are their contributions to what's actually being transmitted to new hosts? So to start to understand these questions, we used a wild-type isogenic tagged strain approach to define this population structure. So what this is, these are all wild-type salmonella that are equally fit. So they're identical, except that we incorporated these 40 base pair barcodes into 2 pseudogenes. So they're identical. There's no differences in growth if you grow it in a lab versus in a mouse. The only difference is they have this barcode, so we can now distinguish these different strains. And so we inoculated mice with an equal mixture of these 8 different strains shown here. And this is our typhoid model of infection. And over 35 days, we also sampled what was being shed in feces, so we can tell what's being transmitted. And at the end, we collected different tissues to define the subpopulation structure. So from this, we can count the total salmonella that's colonizing these different sites. We can also use quantitative PCR to count and differentiate these different barcodes and figure out the abundance of these strains in different sites. And so here's what this data looks like. Here is our equal mixture of this inoculum of these different salmonella strains. In each box, I'm showing you an individual mouse that's housed individually from other mice. And in gray, I'm showing you the abundance of these different strains within systemic tissues. So the systemic tissues are the spleen, liver, and gallbladder. And you can see by eye that the spleen and liver are very similar to each other. And you see that not all mice are also colonized by salmonella in the gallbladder, which is interesting. Furthermore, when you look at the intestinal sites, so here are the proximal sites of the Peyer's patches in the small intestine where the infection begins versus the distal gut sites of the cecum and colon and feces, you can see that different subpopulations do form. And this is just for 4 mice, but we performed this experiment in a total of 19 mice, and we see the same results. And these are just qualitative data, and we wanted to directly measure how different they were. We wanted to quantify these differences. So to do this, we used a common measure in ecology called the Bray-Curtis Dissimilarity Index. And how this works is that for a given site, such as a spleen, you calculate the relative abundances of these different strains of salmonella, and you compare it to the abundances of another site, such as the liver. And you calculate their difference. If the difference is 0, that means these subpopulations are identical to each other. However, if you have a score of 1, this means that these populations are completely distinct. So similar to what we saw by eye, you can tell through these metrics, where each dot represents an individual mouse, that the spleen and liver populations are very similar to each other. However, when you start to look at these systemic sites and how they compare to the cecum, you see that the spleen, liver, and gallbladder are very dissimilar from the salmonella that's populating the cecum. And then furthermore, when you compare the colon and cecum, these are some of the lowest scores observed, meaning this is one subpopulation that's together. And we can also apply this approach now to what's being shed in feces. So we kept track of individual mice, and we can keep track of the strains that are being shed in feces. And what I'm showing you in black here are the different numbers of salmonella strains that are being shed. So this decreases over time in all our mice. So when you first start the infection, about an average of 7 strains are detected in feces, but over time this decreases drastically by day 7, and it's around 2 strains throughout infection. However, at the same time, at day 7 post-infection, there's a dramatic increase in the total of salmonella present. So this represents a clonal expansion. And then we can use the same sort of Bray-Curtis analyses to identify which host tissues are contributing to shedding in feces. And so when we do this, we see the systemic sites are dissimilar from what's being shed in feces. And what is similar are the cecum and colon. So this suggests that the cecum and colon contain important niches for salmonella to be transmitted. However, a limitation of this approach is that we can't keep track of the origin of the strains. So for instance, we have this yellow strain here of salmonella that's dominant in the cecum and colon and feces, but we don't know the route that it took. Did it go from the small intestine to the large intestine, or did it circulate through perhaps the spleen and then receive? We can't tell this by this measure, and so we needed to develop a model where we can mark systemic versus intestinal strains of salmonella. So to do this, we developed a co-infection model, and this involves using, again, identical strains of salmonella, which we can differentiate based on a marker. And we can inoculate with the oral strain, and then we can also inoculate with an IP, or intraperitoneal injection. And how this works is that this strain bypasses the gastrointestinal route of infection and goes straight to systemic sites. So it goes straight to the liver to the gallbladder, and then reseeds the small intestine to be shed in feces. And if we look at the kinetics of a single infection, so if you just infect mice orally, you see that there's an increase in feces that's steady by day 7 throughout. However, if you infect with just a systemic strain, you see that it can be shed in feces as soon as 1 day after infection, but it takes around 10 to 14 days to really stabilize. And so when you inoculate the strains together in a co-infection, what you have set up is you have an established intestinal strain of Salmonella, and then you have an IP strain that's present in systemic sites. And now you can ask, what is the contribution of a systemic strain in the presence of already established intestinal Salmonella? So we perform these co-infection experiments. So we have our isogenic strains here that are identical with their different markers. And over 30 days of infection, we're monitoring what's being shed in feces. So at the end, we can count the total of Salmonella present and figure out how much of each strain is there. And so at the top, I'm going to show you the total numbers of this oral strain, and the bottom will be the total numbers of the systemic strain. And we see that over 30 days of infection, just the oral strain is being shed in feces. We've also reversed the orders of these strains, such that strain 2 is given IP and strain 1 is given orally, and we see the same result. We've seen this over 60 days of infection, and so in all, in 54 mice, we see that just the oral strain is being shed in feces persistently. And so we want to know, well, what happened to the systemic strain? So when we sacrifice the mice, the systemic strain is still present in tissues such as the gallbladder. In fact, it's dominant here, yet we never see the strain being shed in feces. We see that the strain is able to reseed the small intestine. However, when we look at the cecum and colon, we see that it's completely absent and the oral strain is dominant here. So this could suggest that strains of Salmonella that are coming from different sites, a systemic site or an intestinal site, are competing to colonize the cecum and colon in order to be transmitted. However, there are other explanations as to why this could be happening. So a more simple explanation could be that there's just not enough Salmonella that's coming from systemic sites. So it's just a numbers game. But what I was thinking was that perhaps there's interspecies competition between Salmonella and these sites for colonizing the cecum and colon. And this comes from ideas in ecology. What we know with Darwin's naturalization hypothesis is that the more closely related organisms are, the more similar their nutritional and spatial requirements are, and therefore the more intensely they will compete. We also know that priority effects are incredibly important for shaping community assembly. So what this means is that a strain of Salmonella that first arrives to a new ecosystem uses up all the nutrients, takes up all the space, such that Salmonella that comes later no longer has access to the same nutrients and cannot colonize as well. But we first wanted to test this first possibility, and I wondered if increasing the numbers of reseeding gallbladder Salmonella would allow it to be shed in feces. So to do this, we relied upon a gallstone model of infection that was previously established. And this is using a 10-week lithogenic diet. And what this is your standard diet that you get in that animal facility that's now supplemented with 1% cholesterol and 0.5% folic acid. And over 10 weeks, these mice develop gallstones, and Salmonella forms biofilms on these gallstones, and reseeding from the gallbladder is increased by over 1,000-fold. So we applied this to our co-infection model. So for over 10 weeks, and we have our control mice that are on the standard diet, no gallstones. And we have mice that are now carrying gallstones, which we were able to verify in our small animal imaging facility. And you can actually image these gallstones and confirm they have formed before we then perform our co-infection. So after 2 weeks of co-infection, as previously established, there's over a 10,000-fold increase in Salmonella in mice that have gallstones in the gallbladder. And you can look at what type of strain this is. So we are looking at the gallbladder that's completely comprised of the systemic strain of Salmonella. And importantly, we see that increased reseeding is occurring into the small intestine. However, when you look at the distal gut tracts, such as the cecum and colon, you see that the oral strain is predominant here. So this strongly suggests that the presence of the oral strain is excluding systemic Salmonella. So if we thought that this was the case, then we can reason that if you remove the established Salmonella, then you should be able to see reseeding of the systemic strain. So to test this experiment, we have mice that are orally infected with this strain of Salmonella, and we delivered another strain that's isogenic with a different marker that's by the systemic inoculation. And this is a kanamycin-resistant strain. And so what happens is when you treat mice with a single dose of oral kanamycin, this antibiotic only acts in the intestinal tract. It gets rid of all the extracellular salmonella and bacteria that are in the intestinal tract. And you can see this happening as soon as 1 day after infection, and by day 2, all the intestinal salmonella is killed off. However, at the same time, we see a dramatic increase in salmonella that's coming from systemic sites. And so this informed us that 1, salmonella in systemic sites aren't accruing any attenuating mutations that are preventing them from being transmitted. They're perfectly capable of it. It's just that they're being excluded. And importantly, we've been able to identify important virulence factors for this. So the virulence factors I highlighted earlier, hydrogenase and SPY1 and 2, which are important for mediating interactions with the microbiota, are crucial. If you infect orally with these mutants, they're not able to persistently colonize the intestinal tracts, and as they're cleared, then you see an increase of systemic salmonella that's able to reseed. So from this, we've learned some important things about the cecum and colon, how crucial they are for salmonella to colonize in order to be transmitted. And this is a novel form of intra-species salmonella colonization resistance. So what we learned about earlier, the microbiota can mediate colonization resistance, but these are different types of bacteria that are excluding salmonella. Here, we're seeing salmonella from different sites within a single host that are competing for a similar niche. And this has important implications for the epidemiology of salmonella infection, as well as how we treat these infections. So what we know of typhoid patients is that it's actually pretty common for these patients to have gallstones. And a common treatment method is removal of the gallbladder. However, they found that when they've identified the salmonella growing up in these gallbladders, that these strains are ancestral and they're not really being circulated in endemic regions. Furthermore, if you look at what happens to these carriers after they've had their gallbladders removed, over 30 to 40% of them still continue shedding salmonella. So this means that there's another niche besides the gallbladder that's important for salmonella transmission. So now that I've learned how important the cecum and colon are for salmonella to be transmitted, and remembering again that salmonella is not alone in this intestinal community, I wondered if differences in the host microbiota could influence salmonella shedding. In particular, if different compositions could alter salmonella supershedding. And we know that from host to host, we have different microbiotas. This is due to environmental factors, but also genetics are incredibly important. So in the studies of identical twins, these monozygotic twins, they have very similar microbiotas compared to dizygotic or non-identical twins. Furthermore, studies have identified genetically inheritable factors that will shape the types of microbiota you have. That's important for determining, some factors of whether you become lean or obese. And we can also study this in our laboratory system in mice. So the strain that we use are these 129 SVJ mice. This is for our typhoid model, and we know that they have a different microbiota composition than our C57 Black 6 or B6 strain of mice. And there's many different genetic differences between these two strains, but one major determinant is NRAMP. And NRAMP is an antiporter of divalent cations in the membranes of immune cells. So it's crucial for controlling the replication of intracellular pathogens, such as salmonella. And so if you actually infect this mouse that's NRAMP sensitive with salmonella, this mouse will die within 5 days. But we were able to acquire mice from Greg Barton's lab at UC Berkeley that are NRAMP positive. So now we can study persistent salmonella infection in these mice. And there are many other genetic differences that are responsible for the different microbiota compositions. And so I asked if these differences could affect levels of salmonella shedding. And so in this plot here, I'm showing you shedding profiles of 5 different mice of the 129 background. Each line represents a different mouse. And you see that shedding levels reach peak levels around day 14 and are quite stable. You also see the development of supershedders by this time point. In contrast, the shedding profile of C57 and RAMP transgenic mice are incredibly different. You see mice that have somewhat of a showering effect. And so this suggests that salmonella is not as able to effectively colonize the intestinal tracts of these mice. You also see that mice don't become supershedders until late in infection, and they're certainly not asymptomatic. And so this mouse here that became a supershedder earlier at day 21 had incredible cecal pathology. So here is the cecum of a 129 mouse, that's a super shedder. There is some inflammation, but this mouse is completely asymptomatic and does not lose weight. In contrast, this mouse here died shortly after reaching super shedder levels. And so in this B6 transgenic model, we do not have asymptomatic super shedders. And so now I wondered, okay, could this be due to differences in the microbiota that's resulting in these different shedding profiles of Salmonella? And to test this, we have to do some dirty work. So we need to transfer the microbiota of a 129 mouse into that of a B6 mouse. And so these are poop transplants that we performed. And by we, I really mean Denise, more about this picture later, Jens, and Nick, two postdocs in lab who actually performed these transplants. And these instruments weren't actually used in the experiment. How we did this was we treated mice with antibiotics for 3 weeks to wipe out their intestinal microbiota. And then we gave them fresh fecal transplants. So they were oral gavaged with feces from different donors. So as a control, B6 mice are receiving the same types of feces back from another B6 mouse. And then our experimental group is when we have B6 mice that are receiving feces from a 129 mouse. So we can ask if this microbiota transfer changes levels of Salmonella shedding. And so these are our different groups. We allow these communities to stabilize for 4 weeks before we orally infect with Salmonella. And so going over the controls first, so here are the donor mice. These are B6 and ramp transgenic mice, and here are the 129 donors, and their shedding levels are different. And then now I'm going to show you our experimental group. So as a control, these are the B6 mice that received own feces back, and here's our B6 mice that received 129 transplants. And we see that as soon as day 1 of infection, there's significantly more Salmonella that's being shed in mice that receive the 129 microbiota. And in fact, there's no significant difference between these mice and donor 129s. And we see this throughout infection, and we see this at day 21. And importantly, we now see that mice that have the 129 microbiota are able to shed at super shedder levels. And I hypothesized that this was due to the fact that the 129 microbiota is more, facilitates Salmonella colonization of the intestinal tract. And so this is indeed what we see in the cecum. We see that mice that receive the 129 microbiota have similar colonization levels to that of the 129 donors. And intriguingly, we see that there's effects outside of the intestinal tract. So when we look at the spleen, we see that the colonization levels of Salmonella in these 129 recipients are similar to that of the donor. So this led us to believe that perhaps there are systemic effects of having this 129 transplant. And I wondered if this might play a role in shaping the super shedder phenotype. And so what I mean by this is maybe there are roles of the microbiota in modulating tolerance to disease. And so how we measure disease tolerance is looking at a health metric, in this case, body weight, and the pathogen burden. And in this case, the levels of Salmonella that's being shed in feces. And so if an animal or a host is more tolerant, it will be able to maintain more body weight given a higher load of Salmonella. And indeed, this is what we see. So in black are our control mice, and then mice that receive the 129 microbiota are now all super shedders, and they're maintaining similar body weights. And this is a crucial component of the super shedder phenotype. So from this project, I learned that differences in the microbiota composition profoundly affect the shedding of Salmonella. And I wondered if other compositions or alterations to the composition of the microbiota could change Salmonella shedding. And we know that there are some factors that do change this, for instance, diet. A lot of studies have recently identified the important role of diet in both modulating the composition of the microbiota as well as their proximity to the intestinal epithelium. This is work done by Dr. Kristen Earle. She's found that feeding mice a low-fiber diet decreases this distance to the intestinal epithelium and can increase susceptibility to inflammatory bowel diseases. And we've seen with other studies that diets, artificial sweeteners, milk fats will change the composition of the microbiota and predispose hosts to disease. And intriguingly, we also see that in this case, red meat with this altered composition has systemic effects and leads to increased risk of cardiovascular disease. And so I wondered if there were any diets that might increase the super shedding of Salmonella. And in fact, If you go back to this gallstone model of infection, this lab reported that there's actually an increase in fecal shedding due to mice on this lithogenic diet that developed gallstones. So they used a 10-week lithogenic diet, which again is the standard diet that's supplemented with 1% cholesterol and 0.5% cholic acid. And this is confounding in this case because these mice develop gallstones, but I wondered if the diet itself, the high cholesterol present, might increase salmonella shedding. And to test this idea, I then performed these experiments where mice were previously on a base diet for 11 weeks, and then at the time of infection, I switched mice to a lithogenic diet or a high cholesterol diet containing 1% cholesterol. And we can track their shedding, and we see that there's an increase starting at day 3 in these mice that are fed a lithogenic diet or cholesterol diet. And this is significantly increased by day 7. And by day 10, strikingly, almost all these mice become super shedders, when normally it's only 20 to 30%. And so we wanted to know how the diet was modulating this effect. And one thing that we thought about is we know that the diet can decrease the diversity of the microbiota and reduce numbers that are present. And so I wondered if the diet was decreasing colonization resistance. And we know that if we decrease colonization resistance with antibiotics, so pre-treatment of antibiotics, that all mice become super shedders. And wondered if the same thing was happening with our high cholesterol diet. And so to do this, we had mice on high cholesterol diets 2 weeks prior to infection to introduce a disturbance. And then at the time of infection, we removed this high cholesterol diet and returned mice to normal food. However, when we do this, we no longer see an increase in salmonella expansion. And so this indicates that a continuous exposure to the diet is required for salmonella to grow. And so we were interested in figuring out the different mechanisms of how this could be happening. And we don't have time to show all of the data today, but these are some of the possibilities that we've begun to investigate. So it's possible that the diet alters the inflammatory state of the immune system, and we know that salmonella benefits from inflammation. However, when we looked just by gross measures in histopathology, we don't see any differences in uninfected or infected mice. And then if we look at the virulence factors that are required, Salmonella spy 1 and 2, which induces that inflammation, is still required. And furthermore, hydrogenase is also required, which is required for mediating interactions with the microbiota. And then importantly, we wondered if Salmonella was benefiting from the cholesterol in the diet itself. And we performed in vitro growth curves to see if cholesterol added in culture to Salmonella in the lab would increase Salmonella replication. But we don't see this. The cholesterol itself does not enhance Salmonella growth directly, yet it's curious that this continuous exposure to the diet is required. And so this led us to hypothesize that perhaps the diet is changing the composition of the microbiota that is now producing new nutrients or changing the landscape such that Salmonella can then bloom. And actually, we know that the microbiota is incredibly important for cholesterol metabolism. So we know that cholesterol is taken up by the small intestinal enterocytes and is circulated to the liver, and from there can be brought to the gallbladder. In the gallbladder, it's conjugated to glycine or taurine, and these primary bile acids are then secreted into the intestinal tract. And in the large intestine, where the microbiota resides, and the microbiota is responsible for profound biochemical transformations where 2 primary bile acids are converted into over 20 different secondary bile acid forms that we know of. And importantly, the microbiota is also responsible for converting excess cholesterol that's not absorbed into other derivatives. And so I hypothesized that, okay, we're feeding these mice more cholesterol, maybe the microbiota is changing and it's allowing Salmonella to grow. So to be able to identify the different members of the microbiota, we use 16S rRNA gene sequencing. And this is a gene that's present in different types of bacteria. And by looking at their DNA sequences, you can identify the different compositions and you can look for changes. And so for our experimental setup, we have mice that are on the standard diet for 5 weeks, and then they're split into 2 groups. One group stays on this normal diet, and the other half is switched to this high cholesterol diet. And parts of those groups are left uninfected, so now we can tell the difference between changes in the microbiota that are due to just the diet change itself, independent of infection. And then we can also compare the trajectory of changes in the microbiota in infected mice on a standard diet versus a high cholesterol diet. So when we do this sorts of analysis, we are able to look at different abundances of bacteria that live inside of our intestinal tract. And this is just at the class level shown here. And so what I'm showing in this plot here are days 0 through 10 of infection. So day 0 is baseline before I've added any diet or infected any mice. And so they should all be similar. And each bar here represents an average of 5 mice in uninfected mice and 10 mice in the infected groups, and this goes across the 10 days. And so as expected in mice that are uninfected and kept on the standard diet, there's not really any changes at the class level of the members of the microbiota. This is what we expect because we're not changing anything. And then surprisingly, when we infect mice with salmonella and keep them on the standard diet for 10 days, we don't see drastic changes at this level either. However, if we have mice that are on this high cholesterol diet, we see differences in the abundances of bacteria. So for instance, this class here in green, molecules, decreases in mice that are on this lipogenic or in this high cholesterol diet, whether they're uninfected or infected. And we also see an increase of other classes of bacteria, and I'll go more into detail about these later. But this allows us to identify the different members that are present, but again, we wanted to quantify how different these groups are. And so again, we turn to measures in ecology. So this time I'm showing you principal coordinates analysis. And this type of analysis is an unbiased way that will differentiate factors that split up differences between groups. And so this is based on unweighted unifrac distance measures. And so this is actually very similar to the Gray-Curtis dissimilarity index that we learned about earlier. But in addition, this unweighted unifrac also takes into account phylogenetic information, so how similar different types of bacteria are in the gut. So with these metrics, principal coordinates analysis can identify the top 3 variables that explain the differences between groups. And so when we look over 10 days, we have mice that are a standard diet here that don't really change over time. And here we have mice over the 10 days that are put on this high cholesterol diet, and you see that different compositions form. We can do the same for our infected groups, and we see that on both a high cholesterol diet and the standard diet, that infection drastically changes the composition of the microbiota. But we wanted to know how different are they? So mice here that are not super shedders versus super shedders, are they different? And if there's super shedders in this group, are they different from our diet-induced super shedders? And we actually were able to include samples from a separate experiment that was collected after 21 days of infection, and we actually know that differences do form, and you can tell these groups apart. So here are non-super shedders that are on the standard diet, and you see they have composition like this with some Verruco microbiae and some actinobacteria. And then you have super shedders with a different profile. And now with this high cholesterol or lipogenic diet, you see that mice are characterized by a different composition and have more of this Verruco microbiae. So we know that these different communities do arise at the end, but we are more interested in determining, okay, how are these changes occurring and which ones facilitate salmonella expansion? So to get a better look at this, we performed time series analysis of the same principal coordinates analyses. And so here in blue are mice that are on the standard diet and infected. And going across here to the right of the screen is times, so different days. And we see that these groups reach different endpoints of infection, marking different communities. And they also have a different trajectory. And we notice that around day 3 or 4 is when these maximal differences started to arise. And this actually corresponds to our shedding data. So these are mice that are on a standard diet, and you see that around day 3 of infection, levels of salmonella tend to increase in feces before rebounding at day 7. However, mice that are on this high cholesterol diet have a different trajectory, where they increase through day 7 and beyond. So this suggests that differences in the microbiota at these critical time points might be responsible for allowing salmonella to grow. And so we wanted to focus in on these time points. Here I'm showing you baseline, so what these mice look like before we treat them with any diet or infect them. And using statistical analyses, there's no significant differences between these groups. A score of 1 would indicate dissimilar groups. But at day 4, we start to see differences. So we see here that mice, the composition of mice start to separate both by diet. So here's standard diet, and here are high cholesterol diet mice, as well as by infection. And these are different groups that arise. And so now this is ongoing work, and we're still trying to figure out, which members are responsible for these changes and what they're doing functionally. And we have some hints now, but we're still working. We know that some members of the microbiota are decreased in a high cholesterol diet. So one member is neuroplasma, and this is the molecules class that I showed earlier, the bright green, that decreased in mice on a high cholesterol diet. And this is intriguing because neuroplasma is similar to mycoplasma and requires cholesterol to grow. And you would think that if we're giving mice more cholesterol, that they should bloom. But what we actually think is happening is that the cholesterol is feeding another member of the microbiota that's metabolizing it into something else, and this neuroplasma is outcompeted. And so this is evidence of heavy cholesterol metabolism that's ongoing. And we also see these changes where a neuroplasma are decreased in other high-fat diets. Then there's other members of the microbiota, such as Coprobacillus, that are increased in this high cholesterol diet, and not so much is known. This is just the genus level, and the functions of these members of the microbiota could be different based on their specific species as well as strains. But some species of Coprobacillus are involved in secondary bile acids, which is unsurprising because we are feeding this pathway through cholesterol. There's other members of the microbiota that are further increased with this diet as well. This is the Ruminococcus genus. And based on different strains, they can have different metabolic functions. So some strains will be performing secondary bile acids. Other strains are responsible for mucin degradation. So the mucus layer that's on top of the intestinal epithelium where the microbiota is residing. Some of these strains can metabolize mucin and turn them into short-chain fatty acids, which may affect salmonella growth as well as the immune response. Finally, there's other species of Clostridium that we see that are increased in this high cholesterol diet. And these are also reported to be increased in high cholesterol diets in rats and hamsters. So this is not a unique finding. But what's interesting here is that salmonella is increased in, Clostridium is increased in mice that are infected with salmonella, further than mice that are uninfected. And so this could suggest an interesting cross-feeding phenotype where these two members of this member of the microbiota and salmonella are increasing together, and they might be producing nutrients that are allowing both of them to grow up. So we're still working on this, and we're trying to connect differences in composition of the microbiota to what's functionally happening. But we've learned a lot today about the epidemiology of salmonella, how it's affected by diet and ecological forces. So we know that these typhoid patients with gallstones, the epidemiological findings are that the gallbladder isolates of salmonella aren't really being transmitted. And what's interesting is that patients with gallstones actually have increased secretion of cholesterol into their intestinal tract. So this could be a mechanism where, with gallstone carriage, the cholesterol is actually increasing the transmission of an intestinal strain. We also know that the diet profoundly affects shedding levels of salmonella, and so this could be important for what we feed our livestock and preventing food poisoning in humans. And we know that this diet could point out new parameters that are important to be surveilling during outbreaks. So for instance, during an outbreak, we're looking at different food types, but perhaps also the cholesterol content in food. And we've seen that the differences in the microbiota are profoundly shaping a salmonella transmission. And so this is something that we can monitor in the future. And so we know that through genetic differences, human hosts can have differences in the microbiota. And with our mouse work, this could suggest that there might be differences between supershedders and non-supershedders based on this microbiota composition. We know that we can also change this composition with diet. So for instance, Mr. N here, he might have had a different diet and he might have a different microbiota. So there's this idea of a natural supershedder, perhaps typhoid Mariana diet, and a diet-induced supershedder. And we can use and potentially identify biomarkers that can help differentiate hosts that eventually clear infection versus those that become supershedders, and how this is different in natural supershedders versus diet-induced supershedders. And this is important for tracking these carriers of disease. And furthermore, it's interesting to speculate that the reason why Typhoid Mary and Mr. N could have been super shedders in the first place, they both worked in the food service industry, and perhaps it matters what you're feeding someone at the time of infection. So Typhoid Mary, what type of food was she cooking? And Mr. N, if he was serving whole milk, which contains more fat and more cholesterol, this was a factor as well. And at the end of this, we've seen that there's differences in the microbiota, but I think it's really important to try and identify the functional differences. And through these analysis, we hope to identify, say for the high cholesterol-induced super shedding, what nutrients is Salmonella using in this context. And we can hopefully target metabolic pathways for treatment. And we know that antibiotics aren't successful because when you treat an infected typhoid patient with antibiotics, you just increase reseeding of this gallbladder strain. So other strategies need to be employed. Importantly, we've seen that the microbiota is also important for influencing host tolerance, so a dampening of your inflammatory immune responses. And there's a possibility that learning more about these pathways, we can use these immune modulators for treatment of other inflammatory disorders. And finally, in the types of treatments that can one day be provided, we know that diet modification will be important. And if you understand the compositions of the microbiota and what they're producing, if you can deliver probiotics or perhaps engineer resistant microbial communities, this can be really important for controlling disease. Okay. With that, I'd like to thank you all for your attention. I'd like to thank Denise Monak for being a wonderful advisor, and I'd like to thank all of the Monak lab. Nick and Jens, who helped with the transplants, and Amanda is another graduate student who has started working on the B6 transgenic experiments. Sarah and Thibault, who have both been my baymates and have been wonderful to work with, along with the rest of the Monak lab, present members, past members as well. Emily Spulak was a veterinary student that I worked with in the past. I'd like to thank my thesis committee, Manuel, Tad, David, and Justin. Committee meetings are always fun, although this next one might be fun and a little bit painful at the same time. Ami Bhatt for chairing, thank you. I'd like to thank all the Amiga Lab and our joint lab meeting group, Donna and John, Sarah, Stan, and Lucy. Jonathan Hardy for teaching me how to do the small animal ultrasound imaging, the Sonnenberg Lab, the tools, and learning how to do 16S analysis, where I basically knew zero before. So I'm incredibly grateful. Steve as well for some help with mouse work. And then all of the administration and M&I who keep things running smoothly so we can do more experiments and be more productive. Okay, thank you. Questions? Carla?
The super shedders had a new, in that one graph, they both had a new population. I think it was
Gamma proteobacteria? Yeah. So
did that correlation hold up? Or what do you thought?
Yeah, we actually see that happening at the end of infection. So Salmonella is a member of Gamma proteobacteria. And when you have infections like this, there's this idea of like will to like, where if you have an organism that's colonizing and blooming, such as Salmonella, other similar members, species, can thrive in similar environments and also bloom. So I think what we're seeing in these infected mice on this diet might perhaps be more an effect and not so much the cause of what's allowing Salmonella to expand, but we're still digging into that. Yeah.
Sarah.
Lilian, going back to your data, I know you showed us data with using canned for antibiotic work, which it would knock down the intestinal Salmonella as well as some of the microbiota. Did you ever use strep where you're not going to knock down any of the intestinal Salmonella, but you might knock down some of the intestinal microbiota to see if that still plays any kind of colonization role in that systemic thing?
Yeah, what was really interesting about that, so when you treat with streptomycin, you do get the super shedders, but we were able to determine that what's increasing is this intestinal population of Salmonella. So The salmonella strain that we use in these experiments is actually streptomycin resistant. So when you treat with streptomycin, it wipes out the microbiota, but then the salmonella that's already in the gastrointestinal tract blooms. And so that further supports that it's that population in the gut that's expanding. Yeah. Yeah.
So your hypothesis is that there's a effect of the diet in terms of like increasing the amount of superfoods. How does that correlate with like epidemiological set? Like, do communities that have super high fiber diets like that, are they less likely to be superfood? Or is there any?
Yeah, that's something that I'd really like to know more too, and I've been digging in the literature and trying to find that. I think the difficulty is that it's hard to monitor super shedders. So especially for typhoid, the most common sample that's taken is blood. And so we also don't know the shedding levels of most of the patients of typhoid, and we also don't know what their microbiota looks like. It's hard to say. I mean, where we mostly get typhoid, it's not so much in the US, but could be concentrated in Africa and India. So it's hard to compare different diets that way. Yeah, I'm not sure. Yeah, Ami?
So there's a live attenuated vaccine for typhoid that you can take. Based on your research, what diet do you think you would recommend to someone who is taking that oral vaccine?
That's interesting. You would, if it's an attenuated vaccine, you would want that strain to proliferate more in the gut and hopefully produce a stronger immune response, right? And so at this point, I can't say for sure because we don't know what the diet is doing to the immune response. But if you wanted this strain to proliferate, then maybe having a short-term high cholesterol diet just allowed to bloom would be good. And I didn't have time to show this data today, but if you induce a super shedder through this high cholesterol diet and you remove the diet, I still remain these super shedders. And so that could actually be helpful here if you vaccinate, provide the diet, induce these high levels in immune response, then remove the diet so it's no longer affecting the immune response. That could be nice, but at this point, I'm not sure. Yeah. Sasha?
I'm wondering if the super shedders of salmonella, would they be, like both diet-induced and, induced, would they be more likely to super shed other infections, like in the same family, or?
Yeah, that's something that's really interesting in experiments that I have mice for, but haven't done yet. So it'd be really interesting to know how specific this is. You know, if we feed mice this high cholesterol diet, if other enteropathogens such as Yersinia, if they might also expand. So that's an excellent question. Haven't done those experiments yet, though.
Great. Do you have anything else?
That I do. Okay, so first off, I have to thank Denise for being such a wonderful mentor, and she's really let me explore so many different paths in science. And here's, a happy picture of us at graduation, and then here's a very accurate depiction of Denise. So the story behind this is that mental retreat was until he pretty much drew this exact same picture, which is Denise playing tennis and drinking very fine whiskey. No, there's a nice ice cube in there. But this also really shows a lot about Denise, where I'm really amazed by the fact that she can do it all. And I've come to rely on Denise for a lot of science advice and life advice, and I'm just really grateful for having her as a mentor, and I've learned so much in her lab. I'd like to also thank past mentors that I've had. When I was a high school student, actually, I started working in Ali Ban's lab, and that's when I first learned how to make media and slowly culture microbes and slowly learn PCR. That's when I really fell in love with science, and I also would like to thank the grad student mentors that I had at the time. Daniel Keemer, Allison Santoro, and Blythe Layton. And then I'd also like to thank my mentors in the Monarch lab, Tom, Dr. Ruby, who lured me into the Monarch lab, I was also quite afraid of mice when I first started, and Tom showed me how to pick them up after I was, following them around the cage for quite some time. And then there's Smita, who's taught me everything I know about doing mouse dissections and mouse poop. And it's just been so wonderful working with her in lab. We've had so many fun memories, working hard and late together, but it'll always be really fun. And she's just like this crazy encyclopedia of knowledge that I really admire, and I've learned a lot from Smita. And then I'd like to thank the rest of the MONAC lab. These are our current members. We're missing a few. Nina just graduated from the MONAC lab, is heading to med school, but we've had such fun times together. Here we are at MONAC lab retreat, taking a nice selfie in a paddle boat. We dress very well for Halloween and the rest of the time too, usually. I'd like to thank my baymate. So my current baymate, Thibaut. I searched for quite some time to find like more normal pictures of you. You have a lot that have like quite crazy hair, and I love it. And he has a mohawk right now, so very fitting. But it's been so fun working next to you, and every day I laugh. So it's wonderful. And this is my former baymate, Sarah. We were sitting together for 5 years. So many good memories together, so much support. We were known as the poop deck. This came from airmail from Spita. She sent me this grumpy looking zebra, and she gave Sarah these Sour Patch Kids. This is what sustains us. But we've had such a great time working together, and this is just wall art. So Thibaut has this type of fine art in our day, and this is Sarah's source of inspiration. Very enthusiastic manatees, which have been so helpful in grad school. And I've enjoyed my time in this department. You guys are so amazing, and I've had so much fun. I've found some twins. So here's my twin Julie, my twin cat, and the Halloween horse that we made. It's just been such a wonderful time in the department, and you guys make work fun. And then I have to thank my friends from outside department through the med school and Masters of Medicine program, Sarah Prescott. Not have made it without you through that. We learned so much, but I really relied on you for support then. Friends in the CA program and the design school, and Sasha, who's my roommate in Lyman. It's been so wonderful getting to know you and living with you. Every day is just a blast. We celebrate all the best things together. It's wonderful. And then I have to thank my classmates in M&I and also immunology. I found this picture of Jess and Chris and I from 2009. And here we all are, not too long ago. Not too much has changed. But it's just been so wonderful, and you've made Stanford so special. And then I'd like to thank my friends from home. Here we are. I didn't even bring out the high school pictures, but here we are, I think like early in college. And here we are now. It's just been so wonderful. I can't thank you enough for, going through everything together. It really means a lot. Brian too. I've enjoyed getting to know him. He sends me great Snapchats of dog videos all the time. And then I'd like to thank my family, who have always supported me through everything. My grandparents, my Gongong and Papa. My Aunt KK, Uncle Nelson, Auntie Melville, Uncle John, my mom and dad, and then also extended family, so Auntie Rosa and Lap and Olivia. Couldn't find a picture of Derek together, so here he is with Ebola on his Facebook picture, but yeah, I've really learned so much from you guys, and you guys keep me happy and healthy, and I'm just very grateful. More of my family, my grandparents, my grandma and grandpa, that mean so much to me. My grandpa is so inquisitive, and he's always, asking such brilliant questions, and my grandma is the most caring person that I know. They've taken really good care of the family. We eat a lot together, so here we are at Chinese New Year and New Year's at the aquarium. And then I'd like to thank my parents. So this is my dad. Couldn't be here today. He's in Hong Kong. He's a strong guy, obviously. Here is something sweet my mom brought to lab once. She just randomly brought me a card, flowers and chocolate. You know, I feel so lucky for that. And we've had so many amazing adventures in the past. We've gone to places really cold, really hot, and then looking forward to more adventures that we take. I'm so thankful for your support. I owe you guys a lot. Love you guys. Thank you.
So there will be a gathering in the atrium at 4. Hope to see you all there. There will be food and drink.
Try to think So I don't know if I can say